Okay, everyone, welcome to the Mortyn Jones Memorial Lecture. On behalf of the British Society of Endocrinology, we every year host the Mortyn Jones Memorial Lecture to honour Mortyn Jones, who's had a relatively short life, unfortunately, and therefore we commemorate that. And Mortyn, if you don't know the details of him, he trained as a medical doctor at the Royal Free Hospital in London. But although he worked as a clinician, he had a long-standing interest in endocrine research, which he pursued at St. Thomas's Medical School in London. And during his research career, he was well known because he pioneered the use of the in vitro isolated rat hypothalamus for investigation of neurotransmitter regulation of CRF and the site and mechanisms of corticosterone negative feedback. But overall, if we look at Mortyn Jones and his career, one of the key things which we see was how he brought together and integrated and applied his research with regard to clinical medicine. And that brings us very nicely to the award winner of the 2021 Mortyn Jones Memorial Lecture, Waljit Dhillo. So Professor Dhillo was trained as a medic and received a BSc in biochemistry from St. Bartholomew's Hospital at Hospital Medical School at the University of London. He then completed a PhD at the Imperial College London, where he was working on novel neuropeptides and appetite regulation. But notably, he was doing that while he was also working as a specialist registrar in diabetes and endocrinology. So he was bringing together again his research and his clinical practice. He stayed on at Imperial College, where he became a clinical lecturer and consultant. And he passed on to become a reader, and in 2015 he was awarded the prestigious NIHR Research Professorship. Professor Dhillo's research investigates novel aspects of endocrine control of obesity and reproductive function, and today he's going to tell us about translating kisspeptin, neurokinin, dynorphin, neurone biology into new therapies. So with that, I'll hand over to Waljit.
I'll just bring up my slides. Okay, good morning, everybody, or afternoon as it may be in Paris. It's a great pleasure to deliver the Mortyn Jones Lecture. As Neil alluded to, Mortyn Jones combined clinical work and basic science. And I think the basic science discovery that everybody's doing is absolutely crucial into making breakthroughs for patients. And I hope to be able to show some examples of that and share those with you today. These are my conflicts of interest, some consulting work with Myovant, which is relevant for this talk in the later part of the slides, and Can-Do Therapeutics. So we're all familiar with the reproductive axis. I don't need to tell neuroendocrinologists about this axis, it's well established. But the two hormones that I'm going to talk about in this talk are kisspeptin and neurokinin B, and we've heard much about these hormones at previous meetings and at the current meeting. So to take each of these in turn, to start with kisspeptin, again, this comes back to genetic discovery of its importance in human and animal disease, which leads us on to then identification of potential new therapeutic in patients with reproductive disorders. And I'm briefly going to touch on the studies that we carried out with kisspeptin in healthy men and women. I'm then going to talk about our translational work in patients with reproductive disorders, looking at the potential therapeutic effect in patients with hypothalamic amenorrhea, which I'll explain, patients undergoing IVF treatment, more recent work on potential therapy in patients with psychosexual disorders, and some investigator-led studies we've done with a kisspeptin analog in collaboration with Myovant, which would mean that we may be able to translate these findings with a new therapy in the clinic very near in the future. So going back to 2003, these were the two landmark papers that really kicked off the field. And many of you are familiar with these papers. The first paper was from Nicola Daru, and 6 weeks later, a paper from Stephanie Sananara in Harvard and Bill College in Cambridge. And essentially what they showed for the first time using genetic studies, and in the lower paper on animal models, was that animals or patients who had inactivating mutations of the kisspeptin receptor gene, that was the GPR54 gene at the time, as an orphan gene, they failed to go through puberty. And in addition, they had a phenotype of hypogonadotropic hypogonadism. So hypogonadism is pretty obvious. The testes or the ovaries aren't working, and the hypogonadotropic bit is that they had low LH and FSH in the serum. So these two papers have now moving forward with kisspeptin in 2021. We're probably up to about 3,000 papers. So these two papers really kicked off a whole new era of research, which is ongoing and helping our understanding of how the reproductive system works. This slide is, I have to apologize for this slide because it basically is a simplification of probably 2,000 odd papers to say the predominant actions of kisspeptin are through GnRH, so it's upstream of GnRH, acts on the kisspeptin receptor, which is on the GnRH neurons, and then stimulates the rest of the axis. There are receptors in other parts of the axis, and there may be direct effects of kisspeptin, both at the pituitary and both the male and female gonads, but I'm not going to talk about that today. The predominant actions that we see seem to be through the GnRH neurons. So early on, after those papers, we became very interested in this peptide. It seemed to be something that was obviously crucial to human biology. And if a failure of the receptor caused a failure of puberty, the obvious question after the animal studies, which had shown that kisspeptin administration causes an increase in reproductive hormones in animals, was to translate this into humans. So we carried out the first studies back in 2004, and there was the publication in 2005, and administered intravenous kisspeptin for the first time to healthy male volunteers. What you'll notice here is the doses of kisspeptin on the x-axis, and we obviously started very low and moved up, but you'll notice even the 12 is picomoles per kilograms per minute. So this is tiny doses of kisspeptin being infused, and this is the plasma LH on the y-axis, and you can see there's a very marked and large rise in LH, which we were very pleased to see. And importantly, the subjects were admitted to our clinical research facility with heart monitoring, etc., and blood pressure. And at the doses here, and subsequently, that we found no changes in heart rate, blood pressure, or any side effects. And many of you will know that kisspeptin is an endogenous peptide that's raised in pregnancy 7,000-fold. And so, although we were still anxious that this was the first into human studies, it's an endogenous peptide that's raised in pregnancy, and so there were less safety concerns. So this was obviously very pleasing to see this result, and there were more animal studies coming out at the time that kisspeptin was equally as important in women. And so we carried out the first study a couple of years later in healthy women. And what we did was bring healthy women with normal menstrual cycles 6 times to the research facility, twice in the follicular phase, twice in the preoperative phase, and twice in the luteal phase. And in each of the different cycles, they had either saline or kisspeptin. And again, you'll notice here that it's 0.4 nanomoles per kg, so a very small dose. And this was given as a subcutaneous injection, as we'd shown safety with the intravenous infusion. And what you can see here is this is the integral plasma LH after the injection. And what you can see is that in the follicular phase, there's a small but significant rise in LH. Luteal phase, there's a larger rise, but in the preovulatory phase, there's this massive rise in LH. There's a broken y-axis here, which indicates how potent it is in the preovulatory phase. So clearly, kisspeptin can stimulate the reproductive hormones in women, but the hormonal milieu in which it's operating has a big impact on its effects. And this bar here, with the exaggerated effect in the preovulatory phase, will become relevant in terms of the work that we did in IVF treatment. So that was obviously really pleasing to see, but these were healthy volunteer studies, and what we wanted to see was whether we could translate this into patients with reproductive defects. And the first group of women we decided to study were women with hypothalamic amenorrhea. So hypothalamic amenorrhea is the second cause of failure of periods in women presenting to the endocrine or reproductive clinics. And it can be due to low body weight, stress, a number of other factors, and the common factor is that they have a failure of GnRH secretion. So they have normal GnRH, but there's a failure of secretion. And what raised our interest was a beautiful study from Mortyn and Sompere's group, which showed in essentially a rat model of hypothalamic amenorrhea, there was reduced hypothalamic kisspeptin expression. And when they administered kisspeptin, they could restore reproductive axis activity. And so we carried out a study in women with hypothalamic amenorrhea. They were brought into our research facility, 2 cannula inserted for arrival for blood sampling, and then injection of saline or kisspeptin. And these were the results. This is the time after injection of either saline or kisspeptin. This is the LH rise. And what you can clearly see is after an injection of kisspeptin, you get this marked rise in LH secretion, again, without any side effects. So that was great to see. But as many of you will be aware, LH secretion and GnRH secretion causes LH secretion in a pulsatile manner. And it's this pulsatility as well as the amplitude that's important to restore normal reproductive function. So the next question we asked was, could we administer kisspeptin and see whether we could restore some kind of normal LH pattern? So this is the axis switched off in a woman with hypothalamic amenorrhea, and this would be the kind of LH profile that you might see. So the amplitude, you can see the LH pulses should be up like this, and of course, there's no pulsatility there whatsoever, minimal increments there. And the idea is that if we give kisspeptin, would we be able to get this kind of profile where you get this pulsatile GnRH release, which causes pulsatile LH release. So this is the results for one of the women in our study. And what you can see here is on the top left, serum LH on the y-axis and time of sampling on the x-axis. And we sampled the blood every 10 minutes so that we can get a good idea of LH pulsatility. And what you can see here is the red line at the bottom of the x-axis, which is the LH levels are very, very low, and there's absolutely no pulsatility. So when we gave this woman 0.01 nanomoles per kg per hour infusion over that time period, you can see again the very small dose, you start seeing that baseline shift. When we increase to 0.03, there's a significant increase in amplitude. And when we go to 0.1 nanomole per kg per hour, we get this nice pulsatile secretion. Now, we can't confirm that this is exactly the same as an endogenous pulsatility, but this is encouraging. And the proof in the pudding will be ongoing studies, which are going on in our lab at the moment, where we're administering kisspeptin chronically in a pump to see whether we can restore normal reproductive function in these women. Okay, so moving on to another area of research and patient group that we thought would be useful to investigate were women undergoing IVF treatment. And many of you will be familiar with the figures. These are probably even higher now, but 1 in 6 couples suffer with infertility, and probably about 2% of babies, at least in the UK, are born through IVF each year. In terms of the protocol for IVF treatment, it's similar to a normal menstrual cycle, but the idea is that you're overstimulating the ovaries to maximize the chances of pregnancy. So the IVF specialists will, in an IVF cycle, give a woman FSH stimulation to overstimulate the ovaries, produce lots of eggs. Those eggs are immature and they can't be extracted at that point. And what you obviously need is an LH surge to mature those eggs. And we're all familiar with the LH surge in a normal menstrual cycle, lasts about 48 hours and causes nice egg maturation. The problem is recombinant LH has a short half-life. And so HCG is used as a very effective sugar egg maturation in most IVF cycles because it acts directly on the LH receptor. However, it can remain in the circulation for up to 8 days, and then that can cause a problem with excessive stimulation of the ovaries. And that leads to VEGF secretion, and a number of women will suffer with this condition each year, and this is purely iatrogenic due to that HCG administration in an IVF cycle. The VEGF will lead to leaky capillaries and fluid in places we don't want it, and so you'll have fluid in the lungs, pleural effusions, you'll have fluid in the abdomen, ascites, you can get renal failure, and there are a number of deaths each year. And the problem is that HCG, as I said, binds the LH receptor directly on the ovary, and there's absolutely no negative feedback or regulation. And once it's administered, there's nothing you can do apart from watch the woman suffer with ovarian hyperstimulation syndrome and supportive management and ITU management. So what we wondered was, could kisspeptin, because it's acting upstream of GnRH and acting through the person's own endogenous GnRH, you could hypothesize it's a more physiological trigger, because however much kisspeptin you give, there will be a ceiling, because the patient won't be able to release more GnRH than they can make themselves. So the hypothesis was that kisspeptin would be able to give you enough LH for egg maturation, but wouldn't give you excessive stimulation at the ovary, which causes the ovarian hyperstimulation syndrome. So that was the hypothesis. Could kisspeptin be a novel physiological trigger for inducing egg maturation in IVF therapy and be a safer treatment than HCG? I'm not going to talk about this slide in detail. The women that were recruited for this study were essentially women with subfertility that were presented to our IVF clinic. And they essentially were young women with normal endocrine profile and what's commonly kind of termed idiopathic infertility, which means we don't really know why they're infertile, but they're not getting pregnant through normal intercourse. This was the protocol we did, and this is very similar to a normal IVF protocol, but obviously we had a twist with the kisspeptin replacing the HCG. So in a normal IVF protocol, as I said, you'd give FSH to stimulate egg maturation. You give a GnRH antagonist to block a premature LH surge, because that doesn't lead to a good outcome. You need to wait for the eggs to develop. And then at day 11, normally you would give HCG as a trigger for egg maturation. And instead, what we did here was replace that trigger with kisspeptin to see whether we'd still get egg maturation. And then the patient underwent subsequent standard IVF protocol of egg collection, mixed the eggs with sperm, intracytoplasmic sperm injection, and then embryo culture in vitro, and then embryo transfer, and then following up the patients with a pregnancy test. So what do we find? So from an endocrinologist point of view, this was very pleasing to see. These are the two top doses that we used. Kisspeptin trigger here, red is 6.4, blue is 12.8, again, very small doses, nanomoles per kilogram. And that caused a very nice rise in LH, which lasted for the next 7 or 8 hours. And what you can see here is probably the more important, obviously, for the patients, was what happened to the oocyte maturation. And to be honest, we were staggered with these results of how good they were, because this was obviously the first study that had been done. There was 96% of patients had mature oocytes, 91% had an embryo transfer, and we had a number of healthy babies, which was a fantastic outcome for the women in the study. So that was great and really cool, but that wasn't the group of women that it proved concept that kisspeptin could work and be an effective trigger of egg maturation and deliver healthy babies, which is great. But the bit that we're really interested in was, could it be safer than HCG in terms of reducing the risk of ovarian hyperstimulation syndrome that we talked about? And so for the second study, we chose those women specifically. And so we know that women who have a high antral follicle count or a high anti-Müllerian hormone, AMH, those are the women that they've got a polycystic-like morphology or syndrome. Those are the women who will have a higher risk of ovarian hyperstimulation syndrome. And you can see here on the x-axis, the antral follicle count. As the antral follicle count goes up, the risk of ovarian hyperstimulation syndrome goes up. And so what we did was choose women specifically who had a high antral follicle count, as shown here. So these women were very similar to the first group of patients that we studied, but they specifically were selected out because they had a high serum AMH and a high antral follicle count, and would have had a risk, a significant risk of ovarian hyperstimulation syndrome had they had HCG in a normal cycle. And they underwent the same protocol as in the first study, and what we were delighted to see was that there was no clinically significant moderate, severe, or critical ovarian hyperstimulation syndrome. 95 and 98% of patients had no ovarian hyperstimulation syndrome in the early or late phases, and very few patients had mild ovarian hyperstimulation syndrome, for which they didn't need admitting, or very minimal symptoms. And again, we had a number of healthy babies born during that study, which was, again, fantastic outcome for the women in the study. So to summarize this part of the talk, we think that kisspeptin triggering gives us high rates of oocyte maturation. And I didn't mention, we haven't got specific data, but there is data out there that kisspeptin may also have direct effects that are positive on the ovaries and the oocytes themselves. There was no clinically significant OHSS in the high-risk population, which is fantastic. And we had good pregnancy rates, although one needs to take those with caution because the studies are not powered for that. But the numbers that we got look very good, at least as good as HCG. Okay, so and our plan is to try and take this forward again with commercial companies for kisspeptin to replace HCG as a trigger in routine IVF therapy. But those will obviously involve much larger studies and long-term studies. Okay, so changing tack slightly, another area we became interested in at this time was the potential use of kisspeptin in patients with psychosexual disorders. And the reason we became interested in that was obviously there was a huge focus on the kisspeptin GnRH gonadal system, but kisspeptin is also expressed in lots of other areas of the brain. So we wondered what it might be doing there. It's expressed in the human limbic system, and we know that that's involved in reproductive hormone control and behavior. And this is a very busy slide, but it's just to say that the KISS1 gene is expressed in the limbic and paralimbic system, as is the KISS1 receptor. So this gives us an anatomical substrate that the kisspeptin system may have a role there because it's both the ligand and the receptor expressed there. And so the first study we carried out in animals, and this is a study in mice, using a technique called manganese-enhanced MRI. So you infuse manganese into the tail of a mouse. You need a 7 Tesla MRI machine, and then you can monitor the neuronal activity in the mouse's brain. And what we did was give the mouse kisspeptin or saline, and then see what the difference in neuronal activity was in the brain. And what we found, slightly to our surprise, because we were looking at the hypothalamus and the technique can pick up hypothalamic changes, and we'd shown that before using other hormones. What we actually found was that kisspeptin was modulating amygdala activity. And that was a little bit of a surprise to us at that time, because that wasn't very high in the literature at the time. And so we wondered what the significance of this might be. And in a taxi at the BSN meeting back from Edinburgh, I think, I was in a taxi with Kevin O'Byrne from King's, and we were chatting about this data. And as many of you will know, Kevin can do clever things in the mouse brain. And so what he did for us was inject kisspeptin into the amygdala and found a rise in LH. And in a subsequent experiment, injection of a kisspeptin antagonist into the amygdala, perhaps even the more important experiment, that reduced LH secretion and pulsatility. So this suggests that endogenous kisspeptin in the amygdala may have a role in reproductive hormone secretion and neural activity. So the next obvious thing was what would happen in humans. Can kisspeptin modulate human limbic brain activity in human? So we looked at healthy young men using a technique of functional MRI that you may be familiar with. So it looks at the blood oxygen level dependent neuronal activation, gives you a readout of potential neural activity in the brain. And the men underwent a study where they had kisspeptin on one day and then vehicle on the other day, and then we could subtract the two and see whether kisspeptin was having an effect on neural activity in these men. So to activate so the limbic system isn't normally particularly active, and what we're trying to do then in fMRI is activate the system, and you need tasks that the volunteer looks at so that the limbic system will be activated. So they had an emotional pictures task and a faces picture task, which will simulate the limbic system. And they had various pictures shown through to them during the fMRI scan with kisspeptin on board or vehicle on board. And what you can see here is, you have to use your imagination on this one, This is a pornographic picture that was shown. This one is obviously non-sexual couple bonding. This is a negative picture, and this is a neutral picture of a cup. And then for faces, they were shown happy, fearful, and neutral faces. So this would activate the limbic system, and then we would see whether kisspeptin or would be able to alter the neural activity above that in the vehicle group. So this is the protocol: 29 healthy men, randomized, two-way, blinded, placebo-controlled study. And the men came in at time -30 here, had a psychometric questionnaire, and then they had a 30-minute infusion of kisspeptin or vehicle, depending on which day it was, and that was to allow steady state of kisspeptin in the plasma. And then they had an fMRI scan and the picture the emotional and faces picture task that I mentioned, and then a psychometric questionnaire at the end. And what you can see here is the plasma levels of kisspeptin. In red is when kisspeptin was administered, in black is when saline was given. Clearly, kisspeptin administration is going to increase plasma levels in the blood of these patients, these volunteers. An increase in LH as we expected, but no increase in testosterone, which could be a confounder. The effects happen later on. So we had to be quite careful here in pilot studies to make sure that we were infusing to give kisspeptin on board without testosterone increase, which could have confounded the results. So what did we find? What we found was on the emotional task, on whole brain analysis, what you see here is the effects of kisspeptin subtracted from placebo, because obviously there'll be some neural activity in the placebo group as well, because we're giving them the task. But in these areas shown here, there was an activation of kisspeptin above that placebo. And if you do a region of interest analysis as shown here, you can see in a number of areas, the left amygdala, the anterior cingulate and the posterior cingulate, there was an increase in neural activity by kisspeptin compared to vehicle. And what was also interesting, and this is where the psychometric questionnaires were helpful, is that the neural activity correlated with some of the behavioral questionnaires. So for example, here you can see there was negative correlation between hippocampal enhancement by kisspeptin and reward response. And on the bottom one here, you can see an inverse correlation between sexual aversion and putamen enhancement by kisspeptin. And in terms of the emotional tasks, what you can see here, again, is a change, an increase in activation of neural activity in a number of areas shown on the whole brain and region of interest analysis here in the emotional task. And again, there was a positive correlation of change in positive mood with amygdala enhancement by kisspeptin. So we haven't done any studies in healthy females, but many of you will be familiar, and some of you in the audience will be authors on this really neat paper from Julie Backer's group from a couple of years ago, which beautifully shows that the kisspeptin neurons are very important in female sexual behavior as well. So there's no reason to think that similar studies in women will be any different, but we're yet to do studies in women. So this is very exciting. So to summarize this part of the talk, what we've shown is that kisspeptin administration to healthy men enhances sexual and emotional processing, the correlation of fMRI signals with sexual behavior. And the data I didn't have time to show you was that what was really interesting was that these effects were augmented in men with lower baseline reward and lower sexual quality of life. And so this leads on to the next set of studies, which we're currently undertaking in both men and women with psychosexual disorders. And the reason this is important is that it costs a lot of money to treat, estimated $85 million a year in the USA. Current treatments are limited because you need to change the neural pathways rather than Viagra, which is obviously a much more mechanical treatment. And we hope that kisspeptin may have potential as a future safer and more effective treatment for these patients, but further studies are needed, obviously, and some of those early studies we're undertaking as we speak. Okay, so moving on to the kind of later part of the talk. One of the areas that obviously is very important, and we get a lot of questions asked about, is the peptide we're using is the endogenous peptide, kisspeptin 54. But what we need to be able to do is to essentially, as we're trying to translate this, is clinicians around the world will need access to a drug that they can actually use that's a kisspeptin analog. Some of you are familiar with the Takeda compound, TAK-448. They've done a number of studies and then sold the compound to Myovant, and hence the same compound is now called MVT-602. So in the next few slides, where you see MVT-602, it's exactly the same compound as TAK-448, and it is a potent kisspeptin analog. So just to briefly touch on the kisspeptin peptides, many of you are familiar with this. The peptides are a number of different synthetic, a number of different natural peptides. At the top is kisspeptin 54, and it's called 54 because it's got 54 amino acids, and then you've got 14, 13, and 10. And all of these peptides have a C-terminal decapeptide that gives bioactivity. So what Takeda did was modify the kisspeptin 10 form. I won't go into the biochemistry, but it's multiple changes they made over several hundred compounds, and then came up with what was then TAK-448, but is now MVT-602. Some changes in just that sequence, and it's now a 9 amino acid peptide with some sequence changes. But what that does is then give you a very effective compound that's long acting. So these were studies that Takeda had done back in, published in 2014. You can see injection here. These are the doses, very small doses going up to reasonable doses, but still milligrams. And you can see that single injection, really the dose response is minimal. Even if you look at that dose compared to the top dose, if you look at the lowest dose compared to the top dose, there's a huge range, and they're all having a very effective increase in LH in men, and it lasts for up to 72 hours. So this is a potent compound, which is long acting. So we've been lucky enough to do the first studies of this compound as an investigator-led study, with Myovant giving us the compound and our grant funding bodies awarding us to do the studies. And what we've been able to show is basically compare kisspeptin 54, which we used in our studies, and this is the dose we used in the IVS study, for example. So we chose this dose specifically because it would give us an idea of, this is obviously in healthy women, and so that the levels of LH change we're going to get are going to be different, but we knew that this would be kind of a dose that we might use therapeutically. And then we compared it to, we did a dose response, which I haven't got time to show you, but these are two effective doses that we came up with that, again, are very low, 0.01 nanomole per kg and 0.03 nanomole per kg. And what you can see in red here is the effects of, saline is down here in purple, obviously no effect. Kisspeptin causes nice rise in LH as we'd expect, peaking at 6 hours, lasting up to about 12 hours. But what you can see here in the blue and the black lines are the two doses of MVT. You're getting a similar amplitude rise in LH, but a much longer duration of action. And you can see that's kind of coming up to 48 hours, or slightly more. But how is this working? Is this just because MVT's longer acting and sticks around in the plasma for longer and hence combined to the GnRH neurons, or the kisspeptin receptor on the GnRH neurons? Well, that doesn't seem to be the case. So we measured the plasma levels of both kisspeptin and MVT. And what you can see is that the plasma levels go up very quickly and then drop by about 6 hours. They're almost back to baseline. So these prolonged effects that we're seeing with MVT isn't due to an increase in circulating half-life. So we wanted to know, do these compounds have, do these two kisspeptin forms have different actions on GnRH neurons? And at this point, we had a wonderful collaboration with Professor Waljit at the University of Michigan. And what she was able to show with one of her master's students was in their GnRH, mouse GnRH neuronal prep. This is the firing frequency of the GnRH neurons. Control here, treatment just for a few minutes here with either MVT or kisspeptin in red, kisspeptin 54 in red, MVT in blue. And what you can see is kisspeptin has increases firing here, but you can see the action of MVT is much longer. And so this partly explains how MVT is so effective on LH secretion, because it's acting for similar exposure or the same exposure duration to the GnRH neurons. MVT has a much longer pattern of firing. We also looked at two of the commonest groups that cause infertility in women to ensure that the compound will be effective in those groups. So what you've got here is the change in LH after an injection of MVT at time 0. This is, in black, is the healthy controls, and there's a rise obviously in LH as you would expect. In the PCS group, which is the green, the rise is slightly higher. And what's interesting is in the hypothalamic amenorrhea group, just as we've seen with kisspeptin 54, we get a more pronounced rise, and there's almost a biphasic effect, which is interesting. And the FSH response was also very interesting, because in the blue at the top, you can see women with hypothalamic amenorrhea have a really marked FSH rise, and this potentially could suggest that the MVT could be used to stimulate egg maturation in these women, and then later on ovulation. So the other area that I talked about was obviously IVF treatment, and Myovant carried out a study with our input in healthy women undergoing a mild IVF stimulation to see whether it would also be able to stimulate LH secretion during an IVF cycle. And this is what you can see. So injection of MVT at time 0, the right part of the cycle with follicle growth, and you get this nice big rise in LH, which is very similar to what we saw with 54, but it's obviously of a longer duration. And this shows the proportion of patients with ovulation based on transvaginal ultrasound on day 5, and you can see at that 3 milligram dose, 100% of patients had ovulation, which is encouraging. So this is the potential profiles of these hormones with on the y-axis here, serum LH levels or HCG. And what you can see is that HCG, as we've said, acts for too long, but is effective, and that's why it can cause ovarian hyperstimulation syndrome. Kisspeptin is very effective and eliminates ovarian hyperstimulation syndrome. And we think with data I haven't had time to show you, 2 injections 10 hours apart may be more effective than 1, and hence MVT 602, which has a longer duration of action, but acts through the same systems that kisspeptin does on the generation neurone. It may be the ideal trigger for IVF treatment, and obviously further studies are needed, which we're hoping to be able to work with Myovant to carry out in the near future. Okay, so to summarize this part of the talk, which is the main part of the talk, I've shown you that kisspeptin can have potential therapeutic benefit in patients with hypothalamic amenorrhea, patients undergoing IVF treatment to eliminate ovarian hyperstimulation syndrome. Further studies are needed in patients to see the potential efficacy of psychosexual disorders. And it's exciting that there's at least one company out there at the moment, and another that are potentially developing another kisspeptin analog that we'll be able to translate this work into the clinic in the near future. So for the final part of the talk, I'm briefly just going to talk about neurokinin B and some of the studies we've done. And again, it was genetic discovery here that led to its identification of its potential important role in reproductive function. People have been looking at neurokinin B for quite a while before this paper came out in 2009, and the data was a little bit on the fence of whether it was important or not. But this was the seminal paper from Topaglu's group in 2009, and what they showed was that, similar to kisspeptin, if patients had inactivating mutations of NKB or its receptor, patients had hypergonadotropic hypergonadism, very similar to the kisspeptin story, and a failure of puberty. And we know that kisspeptin, neurokinin B, and dynorphin are all co-located in what's now termed the candy neurons in the arcuate nucleus. So this makes a lot of sense. So we obviously were interested to see if neurokinin B administration would affect reproductive function. We thought this would be very similar to the kisspeptin story. And after 18 months of trying, actually what we found, both in healthy men and women, was that neurokinin B administration caused minimal or no stimulation of reproductive hormones. And to cut a long story short, what we think is happening in adults, clearly in normal puberty, NKB is involved in stimulating reproductive hormones and stimulating puberty. But what we think is happening in adults is that there's a high neurokinin B tone, and hence giving more doesn't give you any extra reproductive hormone secretion. The bit at the bottom of the slide, the sentence at the bottom was the one line in this paper: short-lived minimal flushing side effects occurred at higher doses in some of the volunteers. We hadn't really thought too much about that at this point. But actually at this time, there was data from Naomi Rance coming out that perhaps the kisspeptin, neurokinin B, and dynorphin candy neurons are involved in menopausal flushing. And why are we interested in menopausal flushing? Well, this is a major unmet clinical need. 70% of women will experience hot flushes. They can be long-lasting, up to a decade. Current treatments are limited, and as you'll know, HRT is very effective, but a lot of women can't take it because of the risk of breast cancer, strokes, etc. And it's estimated that in the US alone, 20 million women would benefit from new effective treatment. So let's go back to the basic biology, and much of the work I'm going to show you has been done by Naomi Rance in the States over a 2-decade period, and it shows the beauty of basic science translated, giving you new therapies where for the last 50 years or so we've not had any. So if you understand the science, we can move forward and get the right targets for new therapies for patients. So we're all familiar again with the reproductive axis, and menopause. Obviously estradiol levels will fall. There'll be less negative feedback, and you'll have positive feedback on the pituitary and the hypothalamus on generation neurons. But you're also going to have positive feedback on the arcuate neurons. And what Naomi showed 20 years ago or so was actually, this is women on the left, brains of women on the left, the arcuate nucleus, candy population, and women have died in a road traffic accident or something else, and done an autopsy. And what you can see here is women premenopausally, postmenopausally on the right. And what you can see on the red arrows is the women after menopause have candy neuron hypertrophy. So this is the first evidence many moons ago that candy neurons may be important in menopausal flushing. Then moving forward, it's also been shown that if you over-optimize monkeys, you get candy hypertrophy, but when you give them estrogen replacement, that candy hypertrophy goes away. So Naomi produced a really beautiful model of menopausal flushing in the rodent, and what she did was rodents can't tell you they're having a menopausal flush, and so what she did was pick rats which have been over-optimized and set up the system where the tail vasodilatation is a very good correlate of the menopausal flush. And what she was able to do, having established this animal model, was show that ablation of the arcuate candy neurons abolishes this effect. So if we look at our model, we've got low estradiol, we've got less negative feedback and candy hypertrophy. And when you ablate these neurons, the candy neurons at the top, you get rid of the vasomotor symptoms. So this implicates very heavily that these candy neurons are involved in menopausal flushing. So how does this system work? Well, candy neurons project to the median preoptic nucleus, which is the thermoregulatory center, and this actually expresses the NK3 receptor, which is the receptor for NKB. And so if we move forward with our model, we now have low estradiol levels, leading to candy neuron hypertrophy in menopause. There's neurokinin B will be released from those candy neurons, act on the NK3 receptor in the median preoptic nucleus, which then sends signals to the autonomic thermoregulatory pathway, which causes vasomotor symptoms. And so to kind of nail a bit more evidence in favor of this hypothesis, she then administered NK3 receptor agonist into the MPO, and this resulted in vasomotor symptoms. So again, further evidence that this system and this schematic is is the black box essentially that we had for 20, 30 years of low estrogens causing vasomotor symptoms. So we wanted to, so we'd seen in our initial human study that there was some flushing, but that wasn't our primary outcome. So we went back and asked the question, if we do a specific study in premenopausal women who have got low NKB levels in the hypothalamus, could we induce flushing? And what we found, as shown on this slide here, was that volunteers either had vehicle or NKB in random order. And what you can see is when they had NKB in red, 8 out of 10 women flushed, whereas none of them flushed when they had vehicle. And so this added to the literature that NKB, high levels of NKB in menopause may be the cause of menopausal flushing. And in addition, a study came out at that time from Crandall et al., which showed genetic variation in the neurokinin 3 receptor locus was associated with hot flushes. So the hypothesis is that increased NKB signaling causes menopausal flushing. And if you have an NK3 receptor antagonist, ideally oral, then you should be able to attenuate those flushes. And so at this point, I looked around, and there were a number of companies that developed oral NK3 receptor antagonists and had administered them to humans and been used in schizophrenia in early trials, but abandoned because it didn't work. And so I contacted AstraZeneca, and they were happy to give us their compound. And the Medical Research Council funded our first study, which is a randomized, placebo-controlled, double-blind, two-way crossover study in 30 healthy postmenopausal women who were having severe flushes. So this is more than 7 flushes a day, 49 flushes per week. This was the protocol. They had a 2-week baseline period to ensure they were having sufficient flushes for the study. They were then randomized to 4 weeks of treatment with the oral NK3 receptor antagonist placebo, 2-week washout period, which was sufficient because the half-life of the compound is 18 hours. And then they were switched to the intervention they hadn't had for another 4 weeks, and then a 2-week monitoring period. So each patient was their own control, which is quite helpful in these studies because there's a high subject variability in terms of the flushing. And to cut a long story short, what we found was that the oral NK3 receptor antagonists reduced flushing significantly by 45% compared to placebo at 4 weeks, and hot flush severity by a similar amount. Pretty much all of the menopausal flushing parameters that we looked at, which I haven't got time to show you, were all beneficial. So this was fantastic to see. And again, I don't think, to be honest, we would have expected such a big drop in menopausal flushing. And my own hypothesis is that Naomi's discovered the at least efferent pathway that many other pathways may be feeding into that cause menopausal flushing. And just briefly to show you 2 other sets of data from other drug companies which have now taken this forward. This is a study of the Astellas compound that is a 12-week study, very similar patient group to ours, 40 patients in each group. And they did a 12-week study and showed that there's a 50% reduction or so above placebo. And another study with the Bayer compound that was just bought off Candy, which is a once-a-day compound. And this was a 2-week study, but again, showing very similar rates of reduction in hot flushes. Again, about 50% above placebo. So this looks very encouraging. So to summarize and wrap up, neurokinin 3 receptor antagonists could be practice changing. They can very effectively relieve hot flush symptoms without the need for exposure. What's really encouraging and exciting is that larger scale studies are needed, but they're actually underway. So Astellas are ahead of the game with multiple phase 3 studies going on, and the data so far looks very good. And Bayer have just bought out Candy Therapeutics for $800 million to develop their compound. So hopefully we'll have a number of compounds available to treat menopausal flushing. But clearly this could be very useful, and perhaps even more useful in patients with hot flushes due to breast and prostate cancer, where there's androgen and estrogen deprivation therapy lifelong that causes very severe flushing. And they may have other benefits in other diseases that I haven't got time to go into, affecting GnRH positivity and PCOS, etc. So this is, I think, going to be a potentially very exciting area moving forward. So to my final slide, and probably the most important, is the acknowledgements. So the participants who took part in these studies clearly, without their input and time, would be impossible to do. The picture here shows many of the fantastic people who are part of my team that do all of this fantastic work and are very dedicated and a pleasure to lead. And at the bottom are fantastic funders, Medical Research Council, Wellcome Trust, and NIHR. And NIHR in particular have funded me for the last 15 years. So thank you for your attention. I'm very happy to take any questions.
Thank you very much, Waljit, for that fantastic talk. And it's great to see that transition between the basic science and the clinical application. So we've got a couple of questions which have already come through on the question and answer part of the app. So Charlotte Cornhill has asked, do we know where the kisspeptin in the amygdala comes from?
Good question. I mean, we think it's expressed. We know it's expressed there. So, I mean, we're still trying to tease out, and there's many people in the audience who know far more than I do. But yeah, in terms of the exact pathways, I think we've obviously teased out, the basic kind of kisspeptin, GnRH, etc. pathway. But I think there's going to be over the next 10 years a lot of there's already been a number of papers coming out of interactions of the candy neurons with appetite pathways, with behavioral pathways, etc. So I think the easy bit's been done with the first 3,000 odd papers, but actually the interactions, for example, with puberty, there's Mortyn 3 that many people will have read about, and a paper in JCI last year from Ursula Kaiser showing interactions with Mortyn 3, which is a natural inhibitor of puberty, and it may be acting through kisspeptin to have its effects. So I think there's going to be a number of circuits and pathways coming up. And I think the other one that's really interesting is the appetite pathways, because clearly the candy neurons are in the arcuate nucleus, as are many of the appetite pathways. So I think there's going to be a lot of overlap. Okay.
Dave Grattan has said, interesting talk. Thanks, Waljit. I'm interested in the apparent pulse of LH from infusion of kisspeptin.
Yep.
Do you think this is generated in the pituitary or the pattern of the GnRH response?
Brilliant question, as usual, from Dave. That's why I was cautious to be able to say that, I mean, what's amazing is the fact that we're giving this stuff peripherally, and it's acting, how it's crossing the blood-brain barrier, for example, and how it's getting across, and is it hitting the GnRH neurons at median eminence, or is it, doing something intrinsic to the pulse generator? What we think is happening is if you get the dose right, if you go too low, it's just the amplitude. If you go too high, you just get a big burst of LH. And if you go just right, then you get the pulsatility. Whether that's through, the GnRH pulse generator or whether it's through the pituitary, there's not really any way of knowing, I don't think. I think the proof's going to be in the pudding of, the chronic studies. If we can generate that pulsatility and then maintain it, does that kickstart their reproductive axis? And that's going to be the really exciting bit, I think.
He followed that with a second question that was similar to something which I was going to ask you as well, was the sustained LH response to MVT, which you also said was biphasic.
Yeah.
What do you think is happening there? Do you think it's promoting pulsatile GnRH? And then Dave saying, I'm assuming chronic activation of pituitary will be self-limiting. But when you had it wasn't even just a sustained response; it was a biphasic response you got.
I mean, it is. Yeah. I mean, that was specifically in hypothalamic amenorrhea. So hypothalamic amenorrhea is slightly different. I mean, all of the studies we've done with the native kisspeptin 54, we get this, and some people have commented from the papers and have noticed, we get this kind of, it goes up a bit and then it flattens a bit and then goes off again. And MVT is obviously has a different action on the receptor, and we've also got some signaling work with Aylin Panalaglu that we've done in the JCI paper. So they're certainly having different actions. We also know that there's differential sensitivity in terms of tachyphylaxis. So patients with hypothalamic amenorrhea, if you give very high doses, which we did a number of years ago, thinking more was better, it then caused tachyphylaxis and switched off the axis. So I think it's going to be trying to understand in the different patient groups what the doses you need, what the hormonal milieu is. For example, with the MVT, we've also shown in that paper, I don't have time to show you, that if you estrogen supplement normal healthy women, you get a much bigger response, similar to kisspeptin in the preovulatory phase. So I think that's where these detailed studies, I mean, again, it's using the basic science, but in addition, using the clinical models, the actual patient models, to say, you can't, I don't think it's going to be a one size fits all type thing. You have to do the studies in individual patients to get the right treatment. For example, in PCOS, you're going to need more FSH stimulation at the beginning and then LH later on to get the ovulation.
Vincent Prevost, can hot flushes be prevented by aspirin, i.e. is PGE2 release involved in this process? If yes, could neurokinin act on astrocytes?
Wow, that's the answer to that. As far as I know, there's been no studies. I mean, nothing that I've come across that anybody's tried aspirin. So, I mean, if there's potential to use it. I think the menopause studies, you have to be very careful. I mean, I didn't labor it, but you probably saw the placebo effect. So a lot of the rubbish that's out there in the literature has come from there's a big placebo effect that's well documented at 4 weeks and 12 weeks. So there's 25% at 4 weeks and about 50% at 12 weeks, and that's because most of the time, these women don't see doctors and don't have anybody looking after them. And so what you absolutely have to do is a proper RCT, a randomized controlled trial with a placebo control, so that you can see what the real effect of the drug is. I mean, that's the kind of thing that you need to do with the aspirin. It's an interesting thought.
Question from Nicolas Deroux. Great talk. Do you think kisspeptin effect on the limbic system could be mediated via oxytocin release?
That's a great question. I mean, it was shown very early on, actually, that oxytocin, kisspeptin can stimulate oxytocin. So in the original JCI paper in 2017, we actually measured plasma oxytocin levels, and we did it with a group who had a mass spec and were very good at it, and we didn't find a change. So it doesn't necessarily mean it's not, that was in the circulation. So I can't definitively say it's not in the hypothalamus, in the brain. It's certainly a possibility, but we didn't see any changes in plasma levels. We can at least say that.
Okay, and Paolo Giacobini, thanks, Waljit, for that. That was amazing. My question relates as to whether you can control the effect of the NKP antagonist in the POA and ARC, since candy neurons in those areas contribute differently to LH surge and LH pulsatility.
Sorry, can you say the question again?
So can you control the effect of the neurokinin B antagonist in the POA and the ARC, since candy neurons in those areas contribute differently to LH surge and LH pulsatility?
Oh yeah. So that's a good question. I mean, it hasn't been. It's a very good question. It's kind of what I alluded to with the potential treatment of PCOS. So I guess for patients with menopause, we're not interested in their reproductive access at all, because it's not clinically relevant. That's where it's that we could use the NK3 receptor antagonist and whatever it does to a healthy woman. And for example, in the patients with schizophrenia, it lowered their testosterone. The women, they didn't do many studies in women, but you would expect it to affect the GnRH pulse generator. I think what's going to be relevant is, I mean, with an oral antagonist, I don't think you're going to be able to control where it goes. It's going to go where it goes, but you could obviously selectively have ones that are peripherally acting or centrally, but I think it's going to be very difficult to regulate where it goes in the brain. But if you've got, for example, a woman with PCOS, we know, a reasonable number of those women will have upregulated GnRH pulsatility. And so if NK3 receptor antagonists can slow that pulsatility down, that could be, a potential novel therapy for PCOS. So, I mean, there's a number of things where NK3 receptor antagonists are really exciting and potentially useful that we are blocked in doing anything at the moment because the companies are too busy getting their phase 3 studies done and the FDA approval, which is why we've been able to do nothing since, because the company doors are closed. They, breast cancer, prostate cancer are really obvious ones and will make a massive clinical difference. But, the conversations I've had, the companies basically want to get on fair enough, get the FDA approval for menopause, which is a massive market. And then we'll be able to, once they're licensed, be able to look into groups like PCOS and altering GnRH pulsatility, breast cancer, prostate cancer, and potentially others. And there was an animal study in Endo a few years ago with the one of the NK3 receptor antagonists affecting metabolism. So there's, it goes back to the appetite circuits. I think there's going to be a lot of interesting stuff to do over the next 10 years.
Fantastic. Thank you very much, Waljit. That's a great series of questions from our audience as well. So that brings this part of the conference to an end. So thank you again, Waljit, for delivering the Mortyn Jones Memorial Lecture for this year.
Thanks for inviting me.
The next session is there's a lunch informal meeting time for the participants. There's also actually the AGM for the British Society for Neuroendocrinology, and people were sent a separate link in the email from the BSN for that meeting. Thank you very much.